started looking, going back to some of the evidence of the very early physical anthropologists, people started saying, well, okay, these are all Lapita-derived populations. We've got Ni Vanuatu and Kanaks from New Caledonia, Melanesian populations as they were defined by early Europeans, and Polynesian populations here, looking physically quite different as well. So we're getting some disjunct in the genetic data, and similarly in the physiological data. And this has been described as, well, probably the result of a founder effect. What we had was an admixed Lapita population, for example, that had multiple lineages, and due to who got into the canoe to found the new island, you might just get a subset that was not representative of the source population, the parental population. So you might get quite different frequencies of DNA or phenotypic characteristics in different populations. Quite a difficult thing to actually test, but it could be a possible explanation. Well, at this point, we started thinking about, or around as we were starting to collect genetic data, we started thinking about alternative approaches to tracking human migration. And so Lapita people, as we said, introduced a number of plant and animal species when they arrived in near Oceania, and they also introduced those plants and animals out into remote Oceania. So we started thinking that perhaps we could actually look at the DNA of the animals and the plants that people carried with them and identify the genetic history of the origins of those animals, and that might tell us something about the origins of the people, because these animals are only getting around being carried by people. So you figure out where did the animals come from, that's going to be telling you where the canoe came from, which is going to be telling you where the people came from. And so the commensal approach was the approach that I started looking at in terms of trying to reconstruct Pacific prehistory when I started my PhD. And we decided to first study the Pacific rat, one of the 4 animals that was introduced by Lapita peoples. Now, this is Rattus excellens, known as Kiore in Polynesia, or most of Polynesia. It is a different species from the European rats that Europeans brought on their ships, but it is the 3rd most widely distributed rat worldwide. And everywhere we have evidence of Polynesian presence and Lapita presence, we have presence of Rattus excellens. So we decided that was probably the best animal to start studying. Why would people bring rats with them when they settled the Pacific? Well, rat is good food. Here we have some nice big rats along with a little puppy dog, pigs and chickens. We don't generally have to explain why people brought them. And I've tasted it. It does taste like chicken. They breed rapidly and are a pretty reliable source of food. So we do find the remains in archaeological middens throughout the Pacific. They are definitely food remains. So basically, we realized if you could take the rat, get some tissue out of it, extract the DNA, and sequence that DNA, and build the phylogenetic trees, the same kind of family trees from the DNA of different rats, it would tell us about human migration. So that was the topic of my PhD. And then at the time that I was just doing that, technology developed, and in the early 1990s and through the mid-1990s, people realized we could actually get DNA out of archaeological remains. And this really brought kind of molecular archaeology alive. So we could actually get DNA out of archaeological samples, dated archaeological samples, and identify the lineages that were introduced in those founding introductions. So this was one of the studies that we undertook on both the ancient and modern samples, 131 Rattus excellens samples from across the Pacific region, from museums and archaeological sites, as well as a few modern samples as well. And what we found was that there was a remarkable kind of pattern, a phylogeographic pattern of mitochondrial DNA variation in the rats from across the Pacific. We had 3 distinct groups identified. Okay. Group 1, we found only in the Philippines, Borneo, and Sulawesi up here. Group 2, we found through islands Southeast Asia, New Guinea, and out as far as the Reef Santa Cruz Islands, just into remote Oceania. And Group 3, we found only in remote Oceania, with the exception of some samples here in Halmahera. Okay. Just right there, off the bird's head. So what was very interesting is that we found no type 2 rats in remote Oceania. And given the Lepida model of populations moving through here and out into remote Oceania, we would have expected to see that pattern in the Rattus excellens DNA, which we didn't, which was a bit of a surprise. But previously, based on morphological data, if you go back in time, 1935, based on morphology, a scientist had suggested that there was actually 2 introductions of Rattus excellens in the Pacific. One that came through near Oceania into remote Oceania, and another that came around here through Micronesia and into Polynesia. And that was based on morphology alone. What we realized when we went back and started looking at our samples from near Oceania is that almost all the samples that we had came from mainland New Guinea and came from the main islands of the Solomons. And they were not locations that actually had evidence of Lepida archaeology on them. And so we thought, well, maybe we've actually got a sampling problem going on here. So if we're really going to test this and see whether there is a relationship between the near and the remote oceanic rats, we better do some very directed sampling on Lepida sites. So we went back into near Oceania, initially went to Coyle Island, working with some archaeologists who are also here tonight, and trapped rats and looked for archaeological sites. We found no Lepida sites, and we found no Rattus excellens. So that was kind of an interesting pattern that fit with what we expected. But then when we really started targeting Lepida locations, we sampled the islands in New Ireland and off of New Ireland, New Hanover, Manus, and then Matisoo, which has the earliest Lepida site. We didn't actually get to Matisoo, but an island just right off of here. We were invited to, on Emerau, to excavate. We were invited by some local teachers who had heard Glenn Summerhays, the archaeologist I was working with, talking on the radio, and heard him describing Lepida and Lepida sites, and they said, that sounds like this pottery that we're digging up out of our gardens. Why don't you come out and have a look at our island and look for Lepida sites? So we went out there and did some excavations, and indeed did find not only Lepida sites, but some of the earliest, in fact, the earliest dates for Lepida sites that exist at 3,350 BP. Beautiful artifacts, Lepida pottery, fish hooks, arm hands, armbands, shell adzes, the whole shebang. When we started looking at the DNA of the rats from Emerau were still the group 2 rats, the near-oceanic rats, despite the fact that that was the earliest Lepida site. We did find remote oceanic rats out here on a little atoll called Tench, here on New Hanover, and in Manus. So we now have a connection between near and remote Oceania, but what was really interesting is that they were not on the sites where we knew that there was early Lepida. And so it made us started thinking about, well, what could explain this? What was interesting is we started seeing these remote oceanic lineages on locations that had known Micronesian contact and interaction with Micronesia. So maybe we did have something going on here, like Tate originally suggested in 1935. We then started looking at the other Pacific commensals. We did some work on Pacific pigs and the DNA from ancient remains from Pacific pigs, and we found that there was only a single mitochondrial type of Pacific pigs in the Pacific, and out basically all the way through New Guinea and out into Polynesia, and that those pigs were not at all found in Taiwan or the Philippines, but could be tracked back to mainland Southeast Asia. We then started looking at dogs in a major study that we were doing with dingoes and New Guinea singing dogs, but adding Pacific archaeological samples to the study. And we found, in looking at Pacific dogs, there were 3 lineages of mitochondrial lineages of Pacific dogs. We had dingoes and New Guinea singing dogs here. We had 2 different groups of Polynesian dogs, an East Polynesian and New Zealand group out here. And then we had a general Asia and Pacific dog lineage. So we said, okay, let's look at Lepida dogs. What about Lepida dogs? Okay. What do we find in Lepida sites? We started going through all of the early Lepida sites, all the Lepida sites basically through near and remote Oceania, and found almost no dog bone. Found the occasional tooth. So We assumed that dog was a Lapita introduction, but there's almost no archaeological evidence of dog in Lapita sites. However, between 1,000 and 2,000 years ago, dog appears everywhere in large numbers. So there's something interesting going on here between 1,000 and 2,000, starting at 2,000 BP. Now what was very interesting is we've never found any dog bone, archaeological dog bone, in Vanuatu or New Caledonia, two of the islands that you would basically be passing through in order to get to Polynesia, two of the island groups. Okay, we then started looking at chickens, and this is the work of a PhD student of mine, Alice Story, who's now teaching at UNE in the archaeology department. And she was looking at chicken bone and the DNA in chicken bone across the Pacific. She was just starting her PhD when I was contacted by a colleague in Chile when I was there for a conference. And he said he had found some chicken bone in a pre-Columbian site in Chile. Now, chickens are supposed to be a European introduction to the Americas, and he was very excited because he found chicken bones in a pre-Columbian site. He asked if we would do the DNA analysis on them and see if there were possibly Pacific chickens. I was somewhat skeptical, but I agreed. I said that would be quite interesting. Sure, we'll look. And he said, you know, I think Polynesians made contact with the Americas. And we all agreed, yes, of course Polynesians made contact with the Americas, because we have the sweet potato, which is a South American plant that we found in prehistoric, pre-European sites in Polynesia. Earliest is in the Cook Islands, dated to about 1,000 BP. So not only is the plant out there, but the name, kumara, the Polynesian name for the sweet potato, is derived from a Quechuan word, kumar, from a South American word. So it couldn't have drifted over on some raft or being carried by birds or anything else. It had to come with people. Either that or it had a little tag to it saying, call me kumara and plant me in the ground. So we know that there was contact. But I didn't expect that we'd actually find archaeological evidence in terms of the chicken bones. But I thought I'd humor my friend Jose Miguel and we'd do the analysis on it. Well, we did the DNA analysis on it first, and we indeed got the same DNA sequence that we were getting out of Pacific chicken bones. So I thought, okay, that's interesting, but is it pre-Columbian? Well, we got radiocarbon, oh, I didn't have the radiocarbon. We got three of the chicken bones from the site radiocarbon dated, and all three of them came back as being pre-Columbian. They dated from about 1300 to about 1450, and Columbus arrived on the other side of the Americas, of course, in 1492. So we had evidence of Polynesian introduction. So not only did Polynesians get a sweet potato from the Americas, they also introduced. So we had an exchange of chicken for chips, I suppose. So what was very interesting is that when Alice started looking at her chicken DNA, we had two groups that were identified. This was a very early group. We found these in chickens from very early Lapita sites in Vanuatu, 3,000-year-old chickens. And then we had another group up here, a second group of chickens that we found in Polynesia, but not in near Oceania, found in Polynesia, Southeast Asia, and Micronesia. So we're starting to see, again, these Micronesian-Polynesian connections with later dates on chicken. These are the early dates down here. This group here are all dating later. So we've got it in chickens. We're getting this Polynesian